Good morning, everyone. Welcome to MedX channel. Today we are going to talk about anti-CRISPR. Have you ever heard of CRISPR? It is that amazing technology scientists use to edit genes, like cutting and pasting DNA in living organisms. With CRISPR, we have imagined curing genetic diseases, making crops resistant to pests, and maybe even bring back extinct animals. It is one of the most powerful tools biology has ever given us. But what if I told you nature has its own way to turn CRISPR off? Today, we are diving deep into something called anti-CRISPR, tiny proteins discovered in viruses that can stop CRISPR dead in its track. This is the story of hidden biological arm race, the science behind these natural CRISPRs of switches, and why they may be the key to using gene editing safely in the future. To understand the anti-CRISPR, we need first to know CRISPR itself. CRISPR is originally wasn't invented by humans. It was discovered in bacteria. Yes, those tiny single-celled organisms. Imagine bacteria as a little fortress consistently under attack by viruses called bacteriophages, or just phages for short. To survive, bacteria developed a defense mechanism called CRISPR-Cas. So the CRISPR-Cas is the defense mechanism of the bacteria. Okay, here how it works. When the viruses attack the bacteria take a snapshot of part of the virus DNA and store it in its own genome, like a wanted poster. If that virus comes back again, the bacteria use protein called Cas, like Cas9, guided by that stored DNA to cut the virus apart. Scientists realized that if the bacteria can target a specific DNA with Cas protein, it may be we can too. And so CRISPR becomes gene editing revolution, letting us to cut, modify, and rewrite DNA in most of organisms. CRISPRs sound amazing, right? But with this great power, you know the rest. CRISPR can sometimes make mistakes, cutting the wrong spots in the medicine. That is the big problem. What if you had a way to pause CRISPR or to stop it? Something goes wrong. For years, scientists didn't have an answer until they stumbled across anti-CRISPR. Let's discover the anti-CRISPR. In 2013, the scientists studying those bacteriophages, viruses, noticed something strange. Some viruses were winning against bacteria, even when the bacteria had CRISPR defense. A younger researcher called Joseph discovered that some viruses carry special proteins that block CRISPR from working. They named anti-CRISPR protein. Think of this: CRISPR is the lock and key defense mechanism of bacteria. The anti-CRISPR is like chewing gums, stuck in the lock. The key Cas9 just can't work anymore. This was mind-blowing find. It means there is rare natural molecules that could switch off one of the most powerful gene editing tools known to the science. So, how do these anti-CRISPR proteins actually work? There are many types, but the simple breakdown. But here's the simple breakdown. they can block Cas9 protein from binding to DNA. Imagine Cas9 is as a scissor that need to grab DNA before cutting. AntiCRISPR puts safely lock on this scissor. They can stop Cas from recognizing its target, like confusing a GPS so it can find the right address. They can shut down the cutting ability itself. Even if the Cas finds its target, the anti-CRISPR make it useless. No cut, no edit. Scientists have named dozens of these proteins, like ACR2A, ACR2C, ACRF, each specialized for different CRISPR system. Okay, why it is so important to study the anti-CRISPR? It's good science, but why does this matter? Because anti-CRISPR might make CRISPR safer and smarter. In medicine, imagine that we use CRISPR to fix a genetic disease. You only want CRISPR active for a short time. Anti-CRISPR could act as an emergency brake, prevent unwanted DNA to be cut. Agriculture. If you edit crops, anti-CRISPR could prevent those edits from spreading uncontrollably in nature. And in labs, scientists often need precise control over experiments. Anti-CRISPR give them an on-off switch. And finally, in ethics, some fear CRISPR could be misused. Anti-CRISPR could serve as safeguard, making it harder for someone to weaponize gene editing. So, what are the challenges that we can face us in the future? But we aren't there yet. Challenges remain delivering anti-CRISPR protein into cells at the right time and place. Number two, making sure they are turn off the CRISPR you want, not everything. And number three, understanding how viruses may evolve new tricks. Remember, this is ongoing arm race in nature. Many researchers believe anti-CRISPR could be a vital part of the future gene therapy, biotechnology application, and may even policies regulating gene editing. So, the conclusion of this video that we have learned today is that CRISPR gives us the power to rewrite the life code. Anti-CRISPR shows us that nature never leaves such power unchecked. It is fascinating reminder in the science every breakthrough has a counterbalance. In genetics, every tool can have a safely switch. And in the dance between innovation and caution, anti-CRISPR may just help immunity and humanity use CRISPR responsibly. Next time you hear about the new CRISPR breakthrough, remember somewhere there, a tiny protein in virus may be keeping that power in check. Okay, thank you if you lost to this minutes in this video. I hope you get benefit from this video. So don't forget to like this video and subscribe the channel for more videos. Thank you for everything. I love you all. Bye.